Very happy morning, students. Welcome to RGR Academy. This is biology session. Okay, let us begin with the session, the evolution part, the second part of evolution. I hope you're all safe at home and you're doing a necessary practice at home. Before we'll start with this particular chapter, right? The evolution, the second part of evolution. Um, we just have the quick recap of all the things that we discussed before. The previous part, okay? The previous part, if you just have the recap, so we discussed about the first part of the evolution, right? A little bit of the introduction that we discussed, and we discussed about few theories that is like special creation, biogenesis. Then we discussed about the modern theory, right? So these all important areas, the theories that we discussed. After the theories, we discussed about some of the evidences. Evidences like you could say morphological evidences, like anatomical evidence, embryological evidence, paleontological evidence, right? Cytological evidence, like this type of evidences that we discussed almost, and other theories also almost we have done near to that part. When we talk about the other theories, what are the other theories? The Lamarck's theory, the Darwin's theory, mutation theory. These are the some of the theories that we discussed. Okay, almost we try to complete this particular chapter by today. So hopefully by tomorrow, maybe next session, we'll start with human health and diseases. So what we're going to discuss today? Today we discuss possibly about the theories like the natural selection theory, the examples of the natural selection theory. Say like an example, industrial melanism, sickle cell anemia, and we discussed about Hardy-Weinberg principle. So this is also a very important part, the Hardy-Weinberg principle that we'll discuss today, and we discussed about human evolution also that we'll discuss. Start from Dryopithecus, Ramapithecus, Australopithecus, Neanderthal man, Homo habilis, Homo erectus, then Homo sapiens. The substantial stages in the evolution of man that also we're going to discuss. And if time permits, like we also discuss about the mutation. So mutation where we have like point mutation, gene mutation, frame shift mutation, insertion, deletion, translocation, transversion. These are the areas that we'll discuss because the mutation theory, which was proposed by Hugo de Vries, right? So possibly we discussed about mutation if time permits. If time is not okay, pretty well for you because it's a 1.5 session for 1.5 hour session for us. So if time didn't permit for us, then we'll stop with the we won't discuss mutation today. We discuss mutation and molecular basis of inheritance. So we'll discuss about the genetic drift, founders effect, bottleneck effect. These are some important areas, saltation. These are the areas that we're going to discuss today. Hopefully, we try to do the justice for this chapter. Hopefully, we try to end up the stream by 11:30. So let us begin this session. Hopefully, we'll continue with the previous part. So where we are in the other theories, right? So other theories, what are the things that we discussed so far? So in case of like Lamarck's theory, we discussed already, right? Lamarck's theory. Okay, Lamarck's theory, Darwin's theory, mutation theory, modern theory. Now we'll start with the first part, the natural selection theory. So the natural selection theory. Natural selection. Few examples only we're going to discuss as far as the natural selection is concerned, because hope you can recollect the natural selection that we discussed in the last part, right? So population is geometric, whereas the food resources is limited, where it seems to be arithmetic. The struggle for existence that we discussed, and we discussed about the variation in population, that is like continuous and discontinuous mode, survival of the fittest, then inheritance of the useful variation, speciation. All these things are like we discussed already about the natural selection, which was proposed by the Darwin's actually, Darwin's concept that we discussed. Now we're going to discuss about the few of the examples of the natural selection. So what are the live examples, the natural examples? The first example that you take is on industrial melanism. Industrial melanism. So you definitely would have been studying about. So in this question, evolution, you can get two questions. Two questions from this particular chapter. Of course, evolution is a very small chapter, but from this, like you can get two questions. Either you get question from Murray and Miller experiment or Alden and Oparin. That area you can get a question, or you get a question from the human evolution or from saltation or like genetic drift. From these areas you can get a question. So you can expect two questions from this particular chapter, right? So industrial melanism is one of the example, and you take resistance resistance of mosquitoes. resistance of mosquitoes to DDT. These are the very common examples. And sickle cell anemia. Sickle cell anemia. Sickle cell anemia, resistance to microbes, resistance to antibiotics, antibiotics by microorganisms, and we have something called as mimicry also. Mimicry. Okay, these are the common examples, the live examples that we can point out. Okay, so industry, very few things that we're going to discuss today. So if any of these examples come for examinations, like that time you can be very careful. Like these are the examples for the natural selection, in which we're going to discuss about industrial melanism. First, the industrial melanism, this concept actually. So industrial melanism and the micro evolution concept. So can someone tell me, how the industrial melanism, this concept came? This came with the moth species actually. What is the name of the moth? The name of the moth is Biston. Name of the moth is Biston betullaria. Let me write it again so it's not clear. Okay, moth is Western betullaria. One second. Western betullaria. There are two types of moth. One is a gray, other one is a black moth. So Western betullaria. This is the gray moth. Gray moth. And we have the other moth that is called as Western carbonica. Carbonica. This is the black moth. So the concept of this actually. This was first identified. Um, you know we discussed about the mimicry very shortly. We discussed about the mimicry. So here you could see Biston betullaria and Biston carbonica. This happens during the industrialization in England. Okay, this has actually camouflaged. Here you need to study about, for example, this is actually tree bark, the bark of a tree, the bark of a tree. Usually before pollution, what happens? The lichen started growing on the bark of a tree. This, for example, you take this is the lichen. It's what is the lichens, right? So lichen, it started growing on the bark and the surface of the tree. So lichens are nothing but a symbiotic relationship between algae and fungi. So symbiotic relationship between algae and fungi, we call it as a lichens. And lichens, already lichens, which we call it as a pollution indicator. So whenever there is a more of pollutants, more of stools and more of pollutants, definitely what happens, the lichens cannot grow in the polluted area because lichens are sensitive to pollutants. So what happens before industrialization, so before emergence of the pollutants, what happened? There were no more amount of or more number of lichens. So it means there's less number of pollution. So there's less pollution. There was more number of lichens. So for example, you take the color of the lichens. The color of the lichens, usually it is in gray in color. I'm just giving an example, for example, the color of the lichen is gray in color. The color of the tree is a black in color. So for example, black or brown in color, for example, you take. So onto the surface of the lichens, the two types of moths were growing actually. Here you could see one moth. So this is the gray moth. Can you visible is actually the gray moth, and this one is the black moth we are talking about. And these areas, right, these are all lichens we're talking about. These are the lichens. The bark of the tree is completely covered by the lichens. What happened here, the gray moth that we call it as the Biston betullaria has been completely camouflaged or blended with the color of the lichens, so that it couldn't be differentiated. So it has got safeguarded from the predator. It couldn't act as a prey. But in this case, what happens, the black moth was found clearly visible because then it couldn't camouflage with the color of the lichens. So it could actually predated by the predator. So this species started declining actually. So before pollutant actually, before industrialization. But after industrialization, when the pollutants started increasing, what happens? As I already told you, lichens are sensitive to what? Lichens are sensitive to pollutants. Now the amount of the lichens started diminishing or started reducing. Now what happens actually? At this point of time, only the bark of the tree is clearly visible because the bark of a tree is a black in color, where the black moth started camouflage with the bark of a tree, where the gray moth started visible. So it couldn't camouflage. Now, which species started reducing now? The gray moth species started reducing. So this concept is called as, now the population of the gray moth started reducing, whereas the population of the black moth started increasing. So there are two scenarios here: in presence of the pollutants, in the absence of a pollutants, or in terms of, like, that is before industrialization or after industrialization. So before industrialization, what happened? There were more amount of lichens were there. Since there are more amount of lichens, the gray moth can able to survive, whereas the black moth couldn't survive. Whereas in case of, like, after industrialization, right after the pollutants started increasing, the lichens started reduced because the lichens are very sensitive to pollutants. Then what happens? Like, this gray moth couldn't camouflage because the lichens are not there. So automatically what happened? The predators started, like, targeting the gray moth. So the gray moth here, the western betularia started decreasing. This started decreasing, whereas this started decreasing. Western Betularia started decreasing, and Western Carbonica started increasing. So this is one of the classical example for the natural selection. So nature is giving opportunity for both the extremes. Both the extremes were giving opportunity for survival of the gray moth as well as for the black moth in two different conditions. That is before industrialization, after industrialization. This was first identified in Birmingham in the England. Actually, in England it was first identified, and this concept is called as industrial melanism. This is due to the accumulation of the lichens. As long as lichens were there, we could see the prospering of this gray moth. As long as the lichens started diminishing, the lichens started, like, destroyed, then what happens? This gray moth number also, like, started, like, how it got reduced, where this gray moth could been able to easily, like, found by the predators because it couldn't camouflage with the color of the lichens. So by this, actually, the industrial melanism, this comes under the classical example of the natural selection. So if they ask the questions like the name of the moth, the name of the moth is gray moth is a Biston betularia. In case of black moth, it is Biston carbonica. So two different conditions, two different moths of same species, where nature is giving opportunity for the survival of these two different species of the same organisms, so that the nature is selecting either with the black moth, or nature is selecting the, what is that called, the gray moth. So nature is giving equal importance for both the extreme cases. This is a classical example for industrial melanism. Clear, guys? So this is one of the examples that you could see in case of what is it called, the natural selection, that is with industrial melanism. So now we move on to the types of natural selections. Already I gave an example, few examples for natural selection. One example is industrial melanism that we discussed. Sickle cell anemia. Sickle cell anemia, nature usually favors, usually in case of HPA, HPS. So we will be discussing the sickle cell anemia in case of mutation, where we have, like, HPA, HBA, HBS, HBS. In between, there is a stage called as heterozygous condition called as HBA, HBS. This is the actual heterozygous conditions where the nature favors, actually, so sickle cell anemia. So nature also selects the intermediate stage. So that is sickle cell anemia. So sickle cell anemia is again which comes under the classical examples for the natural selection. Okay. So now we move on to the types of the natural selection. Coming to the types of natural selection, there are three types of natural selection. Here we go. This you have studied in the textbooks already. The types of natural selection. There are three types of natural selection. What are three types? Three types of natural selections if you take. The first one example you take. The first example is stabilizing. Stabilizing. The second example you could see progressive. Progressive. And third example that you could take in case of destructive. Destructive. So there are three types of natural selection: stabilizing, progressive, and destructive. I'm just going to give one example under each category so that you can understand what exactly mean by this. Stabilizing. Stabilizing example: sickle cell anemia. Sickle cell anemia. Sickle cell anemia. Project progressive, this can be otherwise called as directional. This is also called as directional natural selection. So directional is other name for progressive. Example, industrial melanism. Just now we discussed about this, right? So industrial melanism. Destructive. Coming to the destructive top, you could say, like, this where the nature eliminates the intermediates. For example, you take snails. Example, snails. that we're going to discuss with an example now. So for example, you take in case of stabilizing. In case of stabilizing example, I told you, for example, you plotted just a graph here. What's happening in case of stabilizing? So in this case, you could see the stabilizing which is happening intermediate. For example, here you could see the graph over here in the stabilizing part. The stabilizing part, you could see this is one extreme and this is another extreme. Okay. So in this case, what happens, like, sickle cell anemia, for example, you take HbA, HbA. Here you take HbS, that is hemoglobin A, and hemoglobin S, both are homozygous actually. But here the stabilizing, where you could see the nature is giving the favorable conditions, where you could see in case of HbA and HbS. So only the nature is favoring. Nature is, like, giving favor for only this type of heterozygous conditions of this type of hemoglobin, that is HbA and HbS, and not for these two extremes. So nature is eliminating these two. Nature is eliminating both the homozygous conditions, the dominant as well as the allele part. Like, this is completely being eliminated on both the extremes. So the nature is favoring. Nature is favoring the intermediate. This is the intermediate form. That is, one is with HbA, other one is with the HbS. So this is an example for the stabilizing type of natural selection. Say, like an example, sickle cell anemia, where the nature is not allowing to prosper both the extremes, where the nature is, like, allowing, nature is supporting the intermediate, that is HbA and HbS. So heterozygous condition. So this is a classical example for stabilizing type of natural selection. Okay. Now we move to the next part, where you could see in case of industrial melanism. In case of industrial melanism, where you could see the directional selection. In this case, you could see the directional selection. It's favoring actually, like, one extreme you could see, one type you could see where the nature is favoring the gray moth, and other extremes you could see the nature is favoring what? Nature is favoring the black moth. So this is, for example, you take the gray moth, and this place you take its example where you could see a black moth. It's a directional actually. Before industrialization, after industrialization, it's a directional actually. Either in this direction or it's going in this direction. So the nature is favoring the extremes, where you could see whether industrialization, before industrialization, the nature is favoring the gray moth. After industrialization, the nature is favoring the black moth. Just now we discussed about the industrial melanism. Yeah, Darwin's finches is also an example that you can take. So the nature is favoring both the extremes, where the extremes were directional. If it is this direction you take, it's supporting the gray moth. If it is going in other direction, it's supporting the black moth. This is an example, we call it as a progressive or directional type of natural selection, where the nature is selecting one particular extreme. Which extreme? The extreme you take, whether it's going for the gray moth, that is before the industrialization, where the progression of lichens took place, it was supporting gray moth. But after industrialization, after the destruction of the lichens, disappearance of the lichens, where the nature started supporting the black moth. So in both the extremes, you could see, you could see in both the directions the natural selection takes place. So this comes under progressive or directional type. So example, you take industrial melanism. So next one, you take disruptive type of natural selection. So this here you could see it eliminates the intermediate. It eliminates the intermediate means, for example, in this case you take it eliminating the intermediate form. So it's eliminating the intermediate form. So this is one extreme. This is the other extreme you can take. So nature is like an appreciating, the nature is encouraging, the nature is selecting the extremes here you could see. The extremes you could see, for example, you take this is a BB, for example, you take this is one type of snail, BB, that's capital B, capital B. This other side you could see the small b, but the nature is not selecting the capital B and small b. This heterozygous condition has not been selected. For example, I tell you, like the capital B, capital B. There are few types of snails which has the ability, to blend with the sand bed. Example, you take the color of the snail and the sand bed, both the colors started blending. So the nature is, appreciating, and nature has started selecting this type of snails. The other type of BB, this type of snail started, growing or blending with the rock, with the color of the rock, because the snail color is also like, the same like that of the rock color. So it started blending. Here the snail started blending with the snail blend, I mean the sand bed. So both the color has got camouflage. Here also the rock color and the snail color has got camouflage. But in between there are few snails, like which couldn't camouflage with the color, so the nature couldn't support this type of intermediate selection in the intermediate varieties. So this comes under the disruptive. Disruptive means where the nature couldn't support, or the nature couldn't take up the intermediate, I mean the homozygous condition. Perhaps it takes the extremes, for example the capital B, that is homozygous condition again. Yeah, the small b, it's again like homozygous condition. It can take, the nature can select both the homozygous, but intermediate, that is heterozygous conditions, couldn't be selected by the nature because it couldn't camouflage, it couldn't be adjusted with the or adapted with the environment. So here the nature is not selecting the intermediate. Example, you take snails for example, because some snails got adapted or camouflaged with the sandbed. Some snails has got adapted or camouflaged with the rock, the color of the rocks. So it couldn't, it got safeguarded, and it couldn't kill by the predator. So here the extremes have been selected by the nature, but not the intermediate, right? So these are the three examples that you can remember. So there are three types of natural selection. What are the three types of natural selection? Stabilizing, progressive or directional type, and destructive type of natural selection. So depending upon the situations, depending upon the scenario where the nature goes on selecting the organisms, either it can be stabilizing, or it can be progressive, or it can be destructive. So other few examples also I can give. So for example, stabilizing selection, like the day-to-day life examples you can take. Stabilizing selection, already we discussed one example in case of stabilizing selection. What we discussed, sickle cell anemia. You know something, sickle cell anemia, the person who's got affected with a sickle cell anemia have less chance of, no chance of getting malaria. So can you justify why? If for example I got sickle cell anemia, I'm the carrier, I got infected with sickle cell anemia. You know what is sickle cell anemia? Because of one particular, like codons, like amino acids get changed, valine to glutamic acid got changed. So because of that, the mutation occurred in my gene, and I mean the particular gene, where the normal structure of the hemoglobin has changed to the sickle shape structure, so it cannot carry oxygen. This is actually the condition for sickle cell anemia. But what the people say is, like, people who got affected with the sickle cell anemia will not get malaria, will not get affected with the plasmodium parasites. What might be the reason for this? What might be the reason? The reason is because of nature. Of course the person has got affected with the sickle cell anemia, but the nature is giving up complement. The nature is giving a supplement stating, like, you got affected with the sickle cell anemia, but you won't be getting malaria in your lifetime. The reason is because it required oxygen. Which requires oxygen? The malarial parasites required oxygen because it usually hosted inside the RBC cells, where what happens, sickle cell anemia, due to the depletion of the oxygen, because the normal shapes change to the sickle shape structure, then what happens, erythrocytic cycle gets stopped. Exactly, then no supplement, no supply of oxygen, because plasmodium, this parasite, requires oxygen for its replication, for the erythrocytic cycle. Since there's no oxygen, because already there is a depletion in what, already there is a depletion of oxygen in the RBC, then definitely what will happen, this plasmodium cannot survive in the sickle shape RBC. So nature is selecting this type of individuals, where sickle cell anemic individuals, giving a complement supplement, you won't get malaria. You need not worry about the malarial parasite, especially the sickle cell anemic individuals. Okay, so this is also a the things that you should remember, where the person you get usually affected with the sickle cell anemia, right? So now you take other few examples for the stabilizing population. One example I already told you, sickle cell anemia. Example you take, short flowers die because of no sunlight. Tall flowers actually die because of the wind damage. But nature is favoring the medium flowers live actually. So there are very short flowers actually. Since there is no light, sunlight penetration, this may die. The nature selection, natural selection I'm talking about as stabilizing, not only sickle cell anemia. In day-to-day life, you could see these conditions also. If the flowers, for example, you take the flowers are having like very short flowers inside. It's deeply buried. For example, these are the small flowers, maybe staminate flowers that you could see. This couldn't be like, where the sunlight could not penetrate. The sunlight could not penetrate inside. So because of this, the short flowers may die. And there are few varieties where you could see for example, you take the very tall flowers, the tall flowers started protruding outside. These tall flowers also may die because of the wind damage. This can also occur. But Which types of flowers where the nature is favoring? The nature is favoring only with the medium sized or medium flowers where the nature is favoring. So this is also one classical example that you can give it for the stabilizing population. And you take babies, you take especially the lightweight babies and overweight babies, heavyweight babies, where the lightweight babies have high mortality rate because they are very weak. They're very weak, and they have they have the heavyweight babies, they have the high mortality rate because when being born. So where the nature is not favoring the lightweight and the overweight babies, but the nature is favoring the average weight babies survive more often, and these babies are seems to be healthy. These are one of the examples. Uh, these are the example. I'm trying to zoom it because sorry, I couldn't zoom it. Okay, so that's what. So I couldn't zoom it, Abhijit. Else I would have zoomed and showed this to you. Okay, fine. So other examples, along with the sickle cell anemia, if you need, I'll send these pictures for you so you can have this for your reference. Okay, flowers and overweight and lightweight babies, where the nature is not favoring the extremes, the nature is taking the intermediate, that is the average weight of babies and the medium size flowers. Okay, so next example you take directional selection. Coming to the next directional selections, I couldn't zoom in. Directional selection. We already discussed about directional progressive. So this is otherwise called as progressive. We discussed already progressive. The classical example that we discussed is industrial melanism. Industrial melanism. Okay. Why, sir, heavy mortality babies have more heavy weight? Because when you compare to like the what is actually the size the baby is supposed to be. If the babies of the chance of mortality, it's not definitely it's a mortality because the body weight seems to be like more of weight compared, for example, 3 kg is supposed to be the normal weight of the babies, right? But the neonates, the newborn babies, it's more than like 3 or 4 kgs or something like that. Definitely, it leads to the accumulation of more of the metabolic substances inside the secondary metabolites started accumulating inside the body, where there are some chances of the babies may go to the mortality rate. So that is why actually the average weight of the babies usually get favored by the nature. Directional selection. Directional selection, we could see, we already discussed about the industrial melanism. Industrial melanism, small horses died, the medium horses also died, but large horses prospered. So it's like an example you take in case of horses, and you could see in case of the small horses died because it's a very disadvantageous. Long time ago we're talking about in terms of evolution. Medium horses also died. This also disadvantageous, but the large horses prospered, and this because of this can well adapted. The food of these horses can well adapted to the terrestrial and different varied arid conditions. So it can run faster and it can well adapt more. The large horses, where the small horses and medium horses couldn't survive better when compared to the large horses. This is one example where you could see in case of directional selection. Other example you take short neck giraffes, medium leg giraffes, and long neck giraffes prosper. This is also this was actually proposed by Lamarck. I hope you can recollect what Lamarck said, because that time use and disuse theory Lamarck which was proposing, there was all the giraffes were having the same sort of the length of the neck. Actually, since the trees are being growing taller and taller, the trees are growing taller, and it started elongating in necks. That is how the giraffe neck started elongated. This is what the proposal of the Lamarck's. But later part, this was disproved by the Darwin, the natural selection, the natural selection, where you could see in this case what happens the lower neck giraffes got extincted, where perhaps the giraffes with the longer neck which can able to catch up those leaves only could able to prosper. This is what actually the confirmation which was given by the Darwin, the natural selection, because nature couldn't select the short neck giraffes and nature couldn't select the uh, medium neck giraffes, but the nature can select only the long neck giraffes alone can be prospered. This was actually given by the Darwin. But the proposal of Lamarck was entirely different. This was the proposal of the natural selections given by the Darwin, where the nature couldn't support the short neck giraffes, nature couldn't support the medium neck giraffes, where the nature can support only the long neck giraffes alone can prosper because the leaves of the tree were reachable. But the proposal of the Lamarck was entirely different. What was this proposal? This was actually the use and disuse. The organs start using and disusing. This was the concept, and actually this started elongating like substantially. The giraffe's necks also start elongated. This is what the proposal was given by Lamarck actually. But later part, this was disproved by Darwin, stating it's a natural selection. It's not just because of the elongation of the neck. It's not just the proposal of the Lamarck's. This because of the directional selection of the natural selection. So this is what actually the contribution that has been given. Along with this, you can take industrial melanism also. Industrial melanism also comes under the category of the directional. Okay, we're done with the second type, the examples. The third type that we'll discuss, disruptive selection. We already discussed the disruptive selection. I told you, like, snails actually. There are three types of snails. One is like BB, that is capital B, capital B. Other one is small b, small b. But the nature couldn't select the intermediate, that is capital B, small b. This couldn't camouflage. Yes, DDT resistance also, exactly, Rubina. The DDT resistance with the mosquitoes variety. This is also again the classical examples for the natural selection. Coming to descriptive, other examples we take. In this case, like in the environment with the black and white rocks, where you could see like white rabbit lives, black rabbit lives, because white rabbit can able to camouflage, black rabbits can able to camouflage, but the gray rabbits dies because it couldn't easily identified by the predator. So the intermediate, that's what I told you, the intermediate couldn't be selected by the nature. This type of selection is called as disruptive selection. How the intermediate, the snails couldn't be selected by the nature because it couldn't adjust, it couldn't camouflage with the surroundings or with the nature. So the intermediate couldn't, selected by the nature. So this is called disruptive. How the rabbits, white rabbits can prosper, black rabbits can prosper because it can camouflage, but the gray rabbits, the intermediate, couldn't, selected by the nature. This is one of the examples that you can remember. And also in the environment, we take, the deep and the shallow flowers you can take. Short beak hummingbird lives, long beak hummingbird also live. How? Because the short beak hummingbirds can get pollen from the flower. The long beak hummingbird also can get pollen from the flower. But the medium beak hummingbirds dies because it couldn't get actually the pollen from the flowers. This is a disadvantage even in could get seen in case of the medium beak amynglus. Camouflage is actually the condition where it can adapt with the environment. For example, you take chameleon. Chameleon, how it changes its color, how it can adjust it or adapt it with the external environment. Camouflaging means even you could see the moth variety that we discussed, Betullaria. How the gray moth get camouflaged with the lichens. How the black moth get camouflaged or adapted with the color of what is it called, the bark of the tree. This is how actually it's adjusting. Okay, okay, it's got adjusted with the surroundings or the environment. You would have seen in the chameleon, the lizard variety, that you could see that how it's getting, even you could see the camouflage and condition kind of mimicries also. Okay, so they can change according to the place. Exactly, it can change according to the, you could see mimicry, you could see aggressive mimicry, the different types of mimicry. Some insects, like, which behave, which act as like it is dead. It can adapt because it means it as if it is a dead, so it can safeguard itself from the predator, because these predators which will not come and eat the dead animals. So what happens, some insects which will be as if it will be behaving like a, as if it's a dead, right? So what happens, like once the predator move out of the place, then the organisms also will move. Like, so this is how actually, like, this is all the strategy, the techniques where the organisms takes by means of camouflage so that it can protect it well from the predator. So this is how actually the camouflage condition means actually. Sometimes what will happen, the insects also will be changing its color as like that of the flower, as like that of the petals, as like that of the sepals, like, corolla, like that it change its color. You have studied about pseudopopulation. These are the, brood parasitism. These are like sometimes, how it's getting camouflaged with the new environment, the conditions. So this is for adaptation, a kind of adaptation where the nature loves this type of organisms. But if it couldn't camouflage, it couldn't adapt with the environment, then the nature couldn't select that type of particular species. So these are the three types of the natural selection. So we discussed three types. What are the things that we discussed? We discussed about stabilizing, progressive, and we discussed about the third type, that is disruptive. These are the three types. Now we move on to the next part, Hardy-Weinberg principle. Coming to the Hardy-Weinberg principle. So what do you mean by Hardy-Weinberg? Sometimes we get questions from this part, Hardy-Weinberg principle. So coming to the next part. So what is the definition for Hardy-Weinberg principle? Definition. It's a relative frequency of an alleles. So relative frequency of an alleles. It remains constant from generation to generation. So generation to generation, where you could see generation to generation in a population. So this is the definition I'm just writing it. So sometimes you get a questions like find out the correct statement. So from generation to generation in a population where organisms are sexually reproducing, where organisms are sexually reproducing. So this is like the definition for Hardy-Weinberg principle. Okay. Okay, fine. You could see in this case. So what's the definition for Hardy-Weinberg? Relative frequency of an allele remain constant from generation to generation in a population where organisms are sexually reproducing. So when this Hardy-Weinberg equilibrium is applicable, that we're going to discuss the equation. So when you talk about the equation, p square plus q square plus 2pq is equal to 1. So p square is the frequency of homozygous dominant and q square is the frequency of homozygous recessive genotype and 2pq is the frequency of heterozygous genotype. Eventually it should be get the value of 1. So if you get the value of 1, the Hardy-Weinberg principle apply. If it's deviated with the 1, then there should be some problem with the Hardy-Weinberg. So Hardy-Weinberg principle usually applied in the branch of population genetics. So in the practical scenario, we may not apply this one, but in the field of population genetics, so if you go with the population genetics, Hardy-Weinberg equilibrium is one of the important tool where you can identify whether it's going to maintain a constant. If from generation to generation, if the population in a population where organisms must be sexually reproducing, this should be have a constant value. So this Hardy-Weinberg comes under the population genetics. So what are the actually the criteria that you need to consider for the Hardy-Weinberg principle? Coming to the Hardy-Weinberg principle, so this is the genetic equilibrium we're talking about, Hardy-Weinberg. So the scenario we're talking about. So scenario is population size is large. So when you start doing the Hardy-Weinberg, the conditions, population size must be large, random mating is occurring, no mutation. Okay, so we cannot say definitely there should not be any mutation. Sometimes a mutation can occur. If mutation occurs, it must be on both sides. It must be bidirectional. Bidirectional, it means both alleles or both organisms or both the population should have the mutation. So if that is the case, it's fine. Or else no mutation. No mutation, but we cannot confirm there should not be any mutation. We cannot say to the tell it to the nature, hey, there should not be any mutation. Definitely there might be some mutation. If mutation occurs, then it must be on both the sides. If then that's the case, no issues with the mutation. If only one population has gone through with a mutation, here is a mutation. Whereas in B population B, there is no mutation at all. Then definitely this Hardy-Weinberg equilibrium, this principle will not work out. If the mutation is going to occur, this is supposed to happen in both A as well as in B, then this you can apply this one for the Hardy-Weinberg equilibrium. And the next condition is no genes are introduced or lost and no selection occurs. It means all genotypes can survive and reproduce equally well. So when the conditions we're talking about the Hardy-Weinberg is where the population must be like big in size, organisms should show the random mating and no mutation. If mutation occurs, it must be on both the sides. This is the third condition. And all organisms have the uniform reproductive potential. All organisms must be having good reproductive potential. For example, you're talking about two populations. One population, that allele seems to be good in reproductive potential, but other population is not at all good, important, or something. It's not up to the mark. Then there's no point in doing this type of the genetic equilibrium. So if you're taking the population, both the population, if you take where both the population must be having a good reproductive potential, then only you'll be getting the value that is constant. If not, then definitely there'll be deviation, and there's no point in doing the Hardy-Weinberg that equilibrium. So there's no point in this. So I'm just going to give an examples like Hardy-Weinberg. For example, the equation says what does the equation says? P square. P square plus Q square plus 2PQ is equal to 1. So now we just derive with the P square, Q square and 2PQ is equal to 1. So when A is equal to P, that we can take. So what does it mean by Q? What is actually the PQ? So if I take the allele, one allele you take, that is A is a P, then you take AA. If you take the AA, this comes P square. How about the Q over here? The Q, which stands for example, you take this, the smaller a. Okay, so then what happens, like, P square, then small a, this will become the Q square. So A stands for P and Q stands for the small a. 2, that is a capital A, that is the P square. 2 small a, that is for the Q square. And for example, you take one small, a capital A and the small a. This will be giving 2PQ. 2PQ. Okay, guys. So 2PQ. So this way I'll remove it. So PQ, then 2PQ. This is P square and Q square. Now I'll just give a value for this one. So for example, I just give a effect of the values of this one. For example, you take in the blood group. You take in case of Mn blood grouping. Mn blood grouping. If they give a value, okay, they just given value for this particular questions. For example, you take Mn blood grouping. If they ask you identify, so what might be the frequency? For example, they given the value for the frequency is 0.7. N, they given the frequency is, for example, 0.3. They asking to identify the Hardy-Weinberg principles. You're going to apply this one. So M and N. So M is what? M is P and N is Q. So you're going to apply this 0.7 and 0.3, that is like P and Q in the equation. So there is the equation, P square plus Q square plus 2PQ is equal to 1. So in place of P, you're going to apply 0.7. In place of Q, you're going to apply 0.3. Mn is actually the type of the blood group. We're talking about the type. We have the different types of blood groups, right? ABO blood grouping, Kell, Duffy, Lutheran. We have the different types of blood group. Mn is like one type of the blood groups that you could see. Very rare blood group, that is a Mn type of blood group. Where you could see M, the frequency of M is 0.7. N, you could see, is a 0.3. The frequency of heterozygous individual. So P is actually M 0.7, Q is actually N, it is 0.2. Then the 2PQ. What is 2PQ over here? 2 into 0.7 into 0.3. So what might be the value now? 2 into 0.7 into 0.3. So it may be giving, like, for example, 0.42. It may be giving 0.42. So now we're going to apply this in this particular formula. So the formula is P square plus Q square plus 2PQ. So what is the P? It's 0.7. It's a P square. Q square is 0.3. And the 2PQ is equal to 0.42. 0.42. So if you sum up everything, so can I get the value for this one? So 0.7 square, 0.3 square, and 0.42. So this gives 0.49 plus 0.9 plus 0.42. So what will be the value now? So this gives the value of 1 approximately. So here the value comes P square plus Q square, 2PQ is equal to 1. So it maintains constant. So like this type of questions also, like it may come sometimes, like they'll ask, can you give any one values, and they'll ask you to identify whether the Hardy-Weinberg principles can apply for this type of one. Okay, yeah, exactly. So many time, not only for Mn blood grouping, any such examples they'll give, alleles they'll take. You take an allele, like the M is one allele and N is another alleles actually. So two contrasting characters that you could see based on the values. If you take based on the values, if you just start incorporating in the equation. So P is what? P is the frequency of allele A in a population. Q, frequency of allele A, that is small a in a population. So P plus Q is equal to 1. But what you should remember is you should remember to apply. So anything, if there is Mn blood grouping, any such thing, they will give actually two different alleles, and you must not apply this into the equation, and so you will be getting the answer. So we're done with this part. And the factors, when we talk about, so when we talk about the factors which affects the Hardy-Weinberg, what might be the factors? Can someone tell me what are the factors usually the Hardy-Weinberg can be affected? What are the factors? The same thing as we discussed already, the criteria, right? So it's 0.09, so it almost will be equal to 1 actually. So if the Hardy-Weinberg principle is applied, then you should get the constant as 1. If the 1, the constant value is not, if the value 1 is, the answer is 1 is not coming, then it means like, the stability is not there. Somehow, somewhere, like alleles or somewhere, like, somehow there should be some mistakes or been would have occurred in the population. Might be like this would have not, you have not taken the criteria or strategies properly. That might be the reason why you're not getting the 1. But Hardy Weinberg, when you take, if you select both the population or alleles correctly, right, as per the criteria, definitely you'll get the value 1. If this p square plus q square plus 2pq is equal to 1, after calculating, if you're not getting the value 1, then it means, right, definitely you could see mutation. Mutation should not be there. That is the criteria. But you cannot tell into the nature there should not be any mutation. Mutation, if occurs, it should occur on both the sides. If that is the case, no problem, then you get the value 1. If mutation is occur only in one allele or one population, no mutation in other population, definitely you will not get the value 1. So it means there should be some problem in the selection of the population itself. Then in that case, Hardy-Weinberg principle, this law will not apply. It's not practically applicable in the day-to-day life situation. But when you talk about, calculating the genetic equilibrium, right? This principle is of like great advantage. Okay, irregular mating, exactly. The reproductive potential that I told you, that is also one of the criteria. If these strategies not properly considered before you going for the Hardy-Weinberg, then there's no point in doing Hardy-Weinberg equilibrium. Then you do better don't do it. But if you are all carefully, you're very keen in maintaining all the criteria that I told you before, then if you do it, definitely you'll be getting the value of 1. If less than 1 or greater than 1, if you're getting this value like, not constant, definitely there is no Hardy-Weinberg principles for that particular population. No stability, no stability. So this you can take in your mind. This Hardy-Weinberg principles can be affected by, how it can be affected? By means of mutation. It can be mutation. Then horse and donkey, yes, that is a mule. We can take an example. Horse and donkey, for example, mule, in case of, if you maintain the equilibrium where you could see, in case of Hardy-Weinberg, you will not get the constant. In case of mule, in case of, you take an hybrid variety, you take a tomato. These are the varieties where genetic modified organisms may not be, you're not getting the constant value. That is 1, you may not get. You get the frequency of, like, the horse and the donkey you take. If you just plot it in a, for example, you take it in a Hardy-Weinberg, definitely you will not get the value of 1. Definitely it will be lesser than 1, or it will be maybe like in sterile, or maybe fertile, or maybe like, you'll not get the value of 1. So only if the stability is maintained, if it fulfills all the criteria, then only you'll get the value that is 1. Then that is genetically it is stable. If it's not, the equilibrium will not be maintained. Okay, what are the factors which affects? Factors: mutation, recombination, gene migration. So these are very important things, like, you should remember: mutation, recombination. Sometimes they'll ask the questions, what are the factors? So mutation, recombination, gene migration, gene migration, gene migration. Then we have a genetic drift. Genetic drift. These are the main factors you could see. Mutation should not be there. If mutation occurs, it should be on both the sides. Recombination, gene migration. Gene migration is of two types: immigration, EMM, immigration, IMM. Immigration and immigration, only the spelling is different. Immigration, EMM, is what? The allele which is going out of the population. IMM is the immigration, the allele which is coming inside. These are the cases where gene migration, it should be balanced actually. For example, this is the population you're talking about. This is the population. I'm talking about the alleles which is going out, the alleles, the population through other means it's coming inside. Then only it will be stabilized. This we call it as, immigration. EMMIGRATION. This is a gene migration. And this one is immigration. IMMIGRATION, immigration. If some certain alleles which is going out of the population, the same number of, like, the amount of alleles supposed to get come inside and should be balanced. If this balancing is not occurring, then definitely this also will affect the Hardy-Weinberg principles. Only the alleles are going out, but the alleles are not coming inside. So no newcomers inside, the alleles are not getting replaced. Then again, the Hardy-Weinberg principles will not apply over here. Okay, so yeah, the gene migration, this should be balanced. Immigration and immigration, EMM and IMM. E means outside, effluent that you got. E means which is going out. I means inside. The alleles are coming inside the population. Both has to be balanced. Only then this principle can be applied. The genetic drift you take. The genetic drift, for example, what is a genetic drift? The natural selection you would have studied about the founder's effect. What do you mean by the founder's effect, guys? Founder's effect. Okay. So the founder's effect. This founder's effect. Okay. The founder's effect. You could see in case of the genetic drift. The genetic drift. The founder's effect. Okay. Coming to this founder's effect. So what do you mean by the founder's effect? The small group will reproduce to populate the new location. Genetic variations will be low. This is not the new population. This all, like for example, you see in this case, there are two species of the same population that you could see. For example, you could see the red beetle. The red beetle, for example, I mentioned is A, and you could see the yellow beetle, for example, the species seems to be B. Both were actually in the same area, same geographical location. But what the founders affect due to the unavoidable circumstances, due to some unavoidable situation, what happens, like, the one particular, a few species of this particular population started, like, moving out of this particular place. For example, you take only the red beetle. The red beetle actually belongs to this population only. On the whole, you could see this is the community, we call it as, this is community one we're talking about. But few species, like, few organisms that start migrating from this original population and started migrating, for example, due to which it couldn't come back to the main own population, own community, but it started adapting to the new environment. For example, you take, this is an island. Now this, for example, this is a male and a female we're talking about. These two beetles which go to the particular island, they proliferate, they adapt, and they adjust with the new environment. Perhaps it's not coming back to its own community. It started developing, started proliferating in the new environment, where for that particular new environment, these two beetles seems to be the founder. So this is called as founder's effect. So who is the founder of the new population? Not someone like who came from the. It's not like alien or someone came from the other planet. The same population, the same community, but it migrated to some other area, the remote region or something, where it started proliferated, it started adapting, and started like adjusting with the new environment, where it started mating, and again the genetic drift started happening over there. This effect we call it is a founder's effect. Why we call it as founder's effect? These organisms are the founder. These organisms are the founder of this particular population. The founder of this population may be anything else, but for this population is concerned. For example, this you take the population two, this community. Now this will start forming a new community. As far as the community two is concerned, who is the founder? These two species are considered to be the founder, are not the ancestors of these two beetles. Only these two beetles are considered to be the founder of the new community. So this effect is called as founder's effect, irrespective of whoever might be the ancestor of this particular population of the community one. But these two particular organisms which move from this area to the remote island, maybe any areas where it started proliferating or it started adapting, exactly, it's a Sewall Wright effect. This we call it as a Sewall Wright effect. This is the founder's effect that we are talking about. So by this, we come to this part, the founder's effect. Okay, guys. So we're done with the founders. And one more thing you need to remember: saltation. I told you already in the last class. Also remember saltation. So what is the saltation? Sometimes you'll get a question from this part. What is saltation? Saltation is something that we call it as a single step large mutation. It's a one step mutation, single step, but it is a large mutation. The single step large mutation, this we call it as saltation. Sometimes you can get this type of questions also, like what is actually the saltation means. So by this we coming to the end part of this bottleneck. I mean the founder's effect. But when you talk about the bottleneck effect, so when you talk about the bottleneck, so this is the bottleneck effect. Bottleneck. So this is just an example. You keep a pebbles, a color pebbles. For example, I placed two types of colors. Like, you could see the red. You could see the red and the blue over here. The red and you could see the blue. So there are two types of population that come in. I mean, two organisms can you see in the same population. So this is a population, original population talking about. This is the original population. Just I am going to tilt this bottle. I'm going to tilt the bottle. Then what happens? Like few organisms which is there near to the neck of the bottle, right? This may be get disseminated. That will be like, this will be transferred, right, to the other area. So it means that's a bottleneck. There'll be drastic reduction in the population. So what are due to the tilting? This scenario may be different, maybe anything, any parameter, maybe a temperature, maybe a nutrient, maybe your any conditions, or extrinsic factor, or intra, or inter specific, anything, maybe like it's a struggle for existence. It may be any reason due to which what happened, the bottleneck would have occurred. Those organisms which is near to the bottleneck, the neck, so it will be forced to move out of the particular population from the original population. Then what will happen? There will be a drastic reduction in the population. Then out of which, okay, if these organisms talking about those who came out of the original population, if this is having a well adaptation, it's well and good. For example, I'm just giving example from the bottle neck. There are like 50 organisms came out of it. Out of these 50 organisms, I'm not pretty sure all the 50 organisms can survive. Out of these 50 organisms, what happens? Only like 5 can survive. All the 45 remaining 45 may not survive. It may die. So the remaining 5 alone can survive. So these are the surviving individuals. The 5 seems to be the surviving individuals. So these surviving individuals can again proliferate. It can mate again. It start going for the next generation. So this is called as bottleneck effect. Today we have class till at we have started by 9:30, only till 11 o'clock. Only till 11 we have a class. So maybe the natural disaster you can take, natural disaster, maybe anything. The scenario may be, if the organisms having the potential to adapt to the new environment, it's well and good, and it go for the next generation. Say like an example that this all the 50 doesn't have the future, then all the 50 will go with an extinction. Then there's no chance of the next generation where the organism will get extincted here itself. Here itself the organism will get extincted. If the organisms will go to the next generation, provided it should have this adaptive ability, so that it can survive even in the worst condition, in unfavorable conditions, so that these organisms can go to the next generation. Okay, so this is called as bottleneck effect. Bottleneck effect, this is the population. This bottle is nothing but it's a population, entire population, original population. The conditions, situations எப்படி வேணா இருக்கலாம். For example, drastic scenario இருக்கலாம், அதுக்கு வேண்டிய nutrients கிடைக்காம இருக்கலாம், maybe like foods கிடைக்காம இருக்கலாம், maybe any reason it might be. Due to the particular reason, due to the particular parameter, it seems to be not favorable. There will be slight shaky nature which occurs in the population. Then what happens? The few organisms are forced to move out of this area. Then there will be reduction in the population. அதுல வெளியில வந்த அந்த பர்டிகுலர் பாப்புலேஷன், ஃபார் எக்ஸாம்பிள் 50 ஆர்கானிசம்ஸ் கேம் அவுட் ஆஃப் தட் பர்டிகுலர் பாப்புலேஷன். ஸோ இதுல ஃபார் எக்ஸாம்பிள் ஆல்ரெடி 500 இருந்திருக்கு. ஸோ 500 minus 50, now the original population will be having only 450 because the 50 organisms had moved out. So now what happens? The 50 organisms has to survive. It's a question mark. Out of this 50, maybe 45 may get destroyed because it couldn't adapt to the new environment. It cannot survive. But the remaining 5 as a chance of survival means it can survive, it can mate, it can reproduce, it can well adapt to the new environment, it can further move ahead, and it can go to the next generation, provided it has the well adaptive nature. Provided even this 5 that doesn't have this ability to get adapted, then definitely what will happen? This cannot go with the next generation, where the next Indian item getting extinction takes place, where this particular all the 50 will go to the extinction. So there's no point of talking about the next generation. So this is actually, we call it as a bottleneck effect. This is otherwise called as population crash. Population crash. This cause adverse effects in the population because there is a reduction in the allele frequency and loss of alleles from the gene pool, and also like it occurs in the random fashion. So definitely there will be reduction in the alleles, reduction in the allele frequency. Definitely this will occurs. Actually, if this occurs in a very small population, again, already the population sizes, for example, it is 500 is fine. For example, you take already the population sizes, for example, you take only 75. Idhila, for example, 50 is moving out of this place, means this will giving a very deleterious effects, very bad effects to this particular population, because already the population is very small. Then if it going with the bottleneck effect with this small population, that is Sewall Wright effect. The Sewall Wright effect, where the speciation occurs, again what will happen? It leads to the it goes to next population, provided the situation is not favorable. Then what happens again? The bottleneck effect failed. Again, the speciation goes to extinction again because it couldn't proliferate again. It couldn't prosper. So again, I don't know, the population will mudinjirum. So it cannot survive. So if it is a large population, somehow the population can manage. But if this occurs in the small population, if this occurs in a very small population, especially severe right effect, the genetic drift occurs, the loss of alleles takes place in a small population, then the population has to face the consequences. The small population has to face the consequences, and definitely it causes the deleterious effects only. So if it is a large population, it can manage, but if it is in small population, definitely it gives abrupt changes. Certain point of time, definitely it gives to a extinction. When if this condition seems to be வெளியில போன ஸ்பீஷிஸால அங்க போய் உயிர் வாழ முடியல அப்படின்னு ஒரு கட்டாயம் வரும்பொழுது என்ன ஆகும்னா கண்டிப்பா சாகும், செத்து தான் போகணும். So in that case what happens, like this leads to a extinction of the species. Okay, population crash, this is called as population crash. Okay, the bottleneck effect. So next we move on to the speciation. So we discuss about the speciation. As far as speciation is concerned, there are two types of speciation: allopatric speciation, and sometimes you'll get a question: allopatric speciation and sympatric speciation. So speciation, speciation. Coming to this part, yeah. Speciation is concerned, gradual. So the speciation may be gradual or abrupt. Two types of speciation: gradual speciation, and we have a abrupt speciation. This gradual speciation can be again classified only allopatric, and we have a sympatric. This is the common classification of the speciation. This may occur by means of the bottleneck effect, the founders effect மூலியமா நடக்கும். Okay, speciation, just now I told you, right? For particular organisms which will migrate from one own population, and this will migrate to the other area, other geographical location, where it proliferates and where it reproduces and multiply, and it forms a new species over there. This we call it as a speciation. So this speciation, we have the gradual speciation, abrupt speciation. So gradual speciation can be classified into allopatric speciation and sympatric speciation. For example, what is a speciation? This is the population talking about A, the one geographical location. Due to the conditions, what happens? A few organisms present over here, they start migrating from this part, that is from population A, and this is migrated to the population. So for example, you take an island. This is an island. Just now we discussed about it. Now what happened? These populations, the few organisms which moved out of this, right? It started proliferating here, and this forms a new species. And this species will become the founder of this population. So this already we discussed the founders effect and the bottleneck effect also. Okay, so this is the speciation. So this is the classification: gradual, abrupt. So allopatric speciation, sympatric speciation. Only one difference that you need to make: allopatric means other place. Sympatric means same place. That is what the main difference that you need to make. Allopatric, different place. This is of, like, for example, you take green beetles. Green beetles is in one place. Okay. For this example, you take A, and this purple beetle you can take in other place. So other places in between you have the bridge, the barrier that you could see in between. The other places, two different places. But sympatric speciation, the speciation of course which takes place, right? But here both the species, both the color beetles will be in the same area, same geographical location itself took place. So allopatric, the population forms a new species after being geographically isolated from its parent population. Example is, you could see the barrier like river, the river which goes in between. Now what happens? The rivers which separates these two types of the species. So allopatric speciation takes place in two different areas. In between there will be a barrier. But in sympatric species, where you could see the small group of a population forms a new species without geographical separation. There won't be any separations. You could see there won't be any of separation. But in spite of that, without any barrier, you could see the proliferations. You could see the development of this two different types of beetles which occurs in the same area. So allopatric and sympatric. Sympatric means same place. Allopatric speciation means it's in the other place. So this you should remember as far as speciation is concerned. So by this we come to almost the end of this speciation part. The last part that we're going to discuss about the human evolution. So possibly try to complete the human evolution, and then I try to close. So you asked me the speciation, right? Someone asked me the speciation. This is the original population. The original population all may be in the same place. After that, what happened? There is some geographical barrier, maybe a river, maybe a mountain, maybe it's anything like the forest. I mean anything that forms in between this between two population. Now this got separated. This is what actually A. Now this has got separated as, for example, A1 and A2. Then after that, what happened? Reproductive isolation. This again turns to, again there will be separations. Now this A1, A2 will be turned to A, and this can be changed to a A2 will be transfigured to a B. Now the new species occurs, for example, A and a B. So this process we call it as, okay, in speciation. In the 11th what we discussed is allopatric speciation, allopatric sympatric speciation that we discussed in the previous slide. Okay, now we'll discuss, we'll start with the human evolution. I'm not going deep into the human populations because another 20 minutes I'm just going to close it. Coming to the man place among the mammals, we'll start with the Dryopithecus. This is only the names that you should remember. In all the criteria and the names, the stages, you should remember at least a few important points. That's the only thing. Especially the cranial capacity, CC, cranial capacity, and also you should remember about the million, how many million years back when the first tool maker, the first and the flesh eating mammals started evolving, right? These are the very important areas. We'll discuss one by one. Few important points are I'm going to discuss. I'm not going to bore you, like, but again teaching in detail. So Dryopithecus, 20 to 25 million years ago. So 25 million years ago. Then comes Dryopithecus first, then Ramapithecus. Ramapithecus is, for example, approximately you take 15 million years ago. After that, you could see Australopithecus. Australopithecus. Australopithecus, 5 million years, right? 5 million years, where the cranial capacity is 600 cranial capacity. Cranial capacity is CC. That is CC, the cranial capacity. MYA is million years ago. Australopithecus, where two species of Australopithecus, Australopithecus robustus, Australopithecus boise. Then comes Homo habilis, Australopithecus, then Homo habilis, Homo habilis. Around 2 million years ago, here the cranial capacity is 680 to 72 cranial capacity. Then comes to Homo erectus. Homo erectus, 1.7 million years ago. So this is the area you should remember. You should remember. Uh, okay, so here you could see, like, when we started, started walking erect actually. So usually what happens at the end of, Australopithecus only, like the man usually would start bending, walking like monkeys or apes. Once we started walking straight, the posture started, coming straight from the Homo habilis itself. So that time itself, what happened? There will be disappearance of the tail. That is the tailbone started disappearing. Okay, so then Homo erectus, Homo sapiens, then we have Homo sapiens, Homo sapiens. Okay, in the Homo erectus only we have the Neanderthal man. So you don't worry about this. We're going to discuss one by one about this. So now we'll start with the first part. The first part we'll discuss. So this is how the evolution which takes place. Okay, first is hominoids. Hominoids. This is if it's not clear, I write it here over here. Hominoids. Then Australopithecus, Australopithecus, then Homo habilis, Homo erectus, and Homo sapiens. So the last form that we got is a Homo sapiens. This the substantial the changes which occurs in the evolution of the man. Now we'll discuss in detail. I mean, characteristics of each and every part of. Yes. So first one is Dryopithecus. Few points and just I mentioned over here. Okay, Dryopithecus, the first form that we're talking about. The life arises in Archeozoic era and Precambrian era. The era is pre. I'm talking about Dryopithecus, Precambrian era. This is considered as the common ancestor of ape and man. Something the question comes like, which is the common ancestor of ape and man? It's a Dryopithecus. The fossils found to be 25 million years ago, the old, and they were more similar to the ape. The first thing that is a Dryopithecus. This is the common ancestors of the ape and the man. It's more like an ape that you could see. Then comes the Ramapithecus. It's so close resemblance with the Dryopithecus and Ramapithecus, almost near to the same. But Ramapithecus is one of the non-human primate ancestors, which forms between 12 to 14 million, 12 and 14 million years ago. The Ramapithecus. This was first discovered by Lewis. Lewis discovered wide curving jaw and hard palate. So curved jaw and the palate, and this was found in the Sivalik hills of India, and later it was named in a Ramapithecus. It was first identified in the Sivalik hills of India, and after that, what happens? This has been after 25 years. Leaky found the similar jars in Africa, and also that it was a contemporaneous like with the Ramapithecus of India. So these creatures are very fewer and fragmentary, and nothing known about Ramapithecus was primitive to Australopithecus. So here the total the blank of like human fossils ancestors are considered. This is about first is Dryopithecus and Ramapithecus. Okay, but apes doesn't have the tail. Then why do I have a tail bone? It's not only you talking about the apes. Actually, there are other species like even the other types of apes that you take. So, monkeys like take the other parts like the other top parts also we have the tail. Maybe we don't know origin. See, that's what I'm telling. This is all hypothesis. Where the evolution we're talking about an hypothesis, we might have the human beings we have been evolved from the apes actually based on the fossils records we're talking about. Okay, but you see, as you said, like being the Homo sapiens have the erect posture, grasping hands, and binocular vision, enlargements of brains, rounding cranium. Yes, it's all correct. That's all the features of the Homo sapiens. So Ramapithecus. Then we started, the Australopithecus, where it started walking erect, Australopithecus where this is called as Lucy. This part you get a questions. Lucy. Lucy is Australopithecus 3.9 million years ago. And more human-like teeth. You got the flat nose and no chin over here. Then bony ridge over the eyes. This is also like the females were very smaller. Females were very smaller than the males actually, and the pelvis and the leg bones resembles those that of the modern man actually. It has got the long cheeks. Australopithecus has got the large cheek teeth, a large chewing muscles. Usually this is got actually. So this is the place where the Australopithecus started acting. The posture started erect. Initially, it was like curved actually, like Ramapithecus, Diopithecus. It was curved. It was walking like apes. Then the posture starting erect actually. You ask me, this is the time where the tailbones that started is not making use of the tailbone. It started the posture that started making erect. So Australopithecus. Then I check with the Neanderthal man. So Neanderthal man, this was found in Europe, East Asia, and the Middle East, and usually the skilled hunters. So they are the Neanderthal man. These people are the skilled hunters. Neanderthal skilled hunters. So and they relied heavily on a meat. Actually, they started consuming the meat also. The meat, and there's like 1,30,000 to 25,000 years back about the thing, and they flourished in the warm periods and in the ice age. In the ice age also, we are seeing this type of species where the Neanderthal man started flourishing. Next comes Homo erectus. We discussed Homo habilis. Coming to the Homo habilis, this important question, guys. Tool maker. Okay. So which species we call it as a handyman? This often you ask get the question. Handyman. They started making preparing their own tools or the tool maker. It's a Homo habilis. So tool maker, the first tool making ancestor. Okay, our handyman. The fossils were discovered by Leakey from Africa. It was first discovered by Leakey from Africa. The cranial capacity. So cranial capacity is 700 cc. So the cranial capacity, how many million years ago back it was discovered, identified, and the features, maybe like the tool maker, first meat eating man. So these are the common features that you should remember. Basically, from these areas only you get the questions, not much deeper in the human evolutions. Only with few important terminologies, few points around that you should remember under each cranial capacity in the million years ago. These are all you should be. Okay, so can you answer the Jafar has a questions where are splint bones present in the horses? So this is again the proposal being talking about Lamarck's theory actually, like he proposed it at that time itself. There'll be the emergence of the splint bones in case of like the horses itself, where later part this was again disproved by the Darwin's actually in the natural selections. Can you tell me, guys, where exactly the splint bones were present? Usually in the neck area. Usually in the neck area, the giraffes and the other places where, especially in these animals, where you could see the necks you could see the splint bones. Yes, mostly in the neck you could see the splint bone. Okay, Homo habilis, then Homo erectus. Coming to the Homo erectus, here the clear presence in Africa, a clear presence in Africa that you could see, and some researches between Africa specimens and the Asian specimens that you would have been identified. And here in this case, you could see the increased body size and robustness, similar adaptive pattern, changes in the limb proportions, and greater encephalization. So these are like certain things like which all they started behaving as if like a human being, where like the consciousness and the intellectuality, like these all started like increasing. So the homo habilis, the cranial capacity is 612 exactly. So 612 is a, see guys, even if I'm not mentioning it also, oh, you're doing it, cranial capacity and how many million years back, these two pieces of information are quite essential for the human evolution, right from the Dryopithecus till the Homo sapiens. So Homo erectus. Then comes the last Homo sapiens. Homo sapiens, we call it as a knowing man. This 130,000 years present and found worldwide. Highly crafted. They started using highly crafted tools and everything. And agriculture after this, like this started emerging. And then it's omnivorous in diet and complex social organization. So this was actually called as Cro-Magnon man. This was Cro-Magnon man that lived in Western Europe during the late Ice Age also. Okay. Cranial capacity is actually like, as long as the evolution started appearing, like, the cranium, that started, reducing in size. When you see in the initial part, when the skull size seems to be very bigger. But later part, what happens, like, right from the Dryopithecus, when it reaches the Homo sapiens, the cranium, the skull part started reducing in size. They started using a brain and they started, like, doing it intellectual by making a tool making, the agriculture, like civilization. These are all the different areas that started improving. Then automatically, what happened? The size of the skull or the cranium started reducing in size. So based on this, we calculated the cranial capacity. Initially, it was a Dryopithecus. You see the big head, the jaw and all bigger in size. Usually it was like that. Later, but evolution it takes place. What happens? The cranium, the size of the cranium started reducing. It started reducing in size, and also the intellectual quotient, IQ level also started. They started differentiating what is right and what is wrong. Started wearing a dress. These are all the common manners. Toilet making facilities. The tool making facilities, dressing themselves. Like these are all the other areas. Like substantially, they started improving. This is because of the cranial capacity. This occurred in case of human beings. Later, in case after the Homo habilis, Homo sapiens, these all things which started occurring. Okay, so only right from the Neanderthal man, started from the Neanderthal man, the woolen sweaters, like these are like after hunting, like these, they started thinking, why don't we make use of this, the wool, right, fur as a cloth for us to protect our body, to cover our body. Initially, our ancestors in the very beginning stage, they were not wearing dress and everything. After that point of time, after cranial capacity started improving, then started wearing a dress. They started covering the body. They started differentiating the male and the female. These all differentiations started occurring actually. But initially, in the point of Dryopithecus and that areas and all, we are more like monkeys, and we were like loitering here and there in the forest where we don't know to differentiate what is right and what is wrong and everything, right? So this is what the cranial capacity started improving. And nowadays, if you talk, we started designing a robots. We started reporting, making a computers, like many and achievements that we're doing. This is because like IQ levels start increasing, and we started equipping our mind in such a way to do that. Okay, fine. So we're done with this part, that is Homo sapiens. So almost we're done with the human evolution. The last part that we're going to discuss, human evolution, we have done. I'll check if any other part I missed out. So these are all actually the images I have forgotten to show you. This is actually the Lamarck's actually. Lamarck's theory I told you, right? But later it was disproved, but I forgot to show this image for you. Lamarck was explaining how this elongation of the giraffe's neck which upward. Initially it was, eating the, the shrubs like small plants. Later, like, the trees started like growing and increasing in size. It started elongating in necks. And this is how the giraffe started appearing. But later, but it was disproved by Darwin. What the Darwin said actually, those species who couldn't adjust with the new environment, these species got extincted. This species got extincted. Only those species which can adapt with this type of environment, the nature selected only this type of giraffes. All other older types of giraffes have been extincted. This proposal was given by Darwin. But Lamarck's theory was entirely different. Lamarck's theory was based on use and disuse, based on the use and disuse. But later part, this giraffes, the elongation of the neck, this concept was also ruled out by Darwin. The same way where you could see in case of horse also, Lamarck tried to prove actually. Initially, the type of horses, what is it called? Eophus, Oligoephus, then Merychippus, then Pleioephus. Now only you could see the modern horse actually. So again, what happens, like this was disproved by the Darwin, actually, those species like which couldn't adapt to the new environment, the nature couldn't select these species has become extincted. Only the modern horses only like which couldn't survive in the different environment. Okay, speciation originated from the same population. Might be yes, of course, like it might have been from the same population. So then what happens, like it would have been migrated to the different areas. Then only the speciation started occurring. Initially, the Australia also like was a part of Asia. Later part only with Australia got separated, right? The same way, all the organisms were being gathered in one particular area. Due to the circumstances, due to the different scenario situations, what happened? One particular species of like the particular population started migrating to the other area, and they started proliferating, and they start founding a new species, a new environment over there. They become the founder of that particular area. Initially, they would have been from the particular the same area only. Okay, from the same population. It's not compulsory; it should be of the same population. If there are situations where you could see maybe from the different populations also, there are some possibilities out there. Okay, okay, okay. So we done with this part. That is yeah, before that actually as we talk about before the areas like we think like the giraffes like we have the horn like structure. These are the evolutions of like when you talk about the previous parts, we discuss about different types like even the giraffes, the horn and all the structures. But during the course of evolution, but especially when you talk about the leg part this started adapting with the new sort of environments. So this is how actually it occurs in the time of we assume this is how the modern horses would have been evolved. Okay, so this by done with the part. So the last part I just close it by means of mimicry. So what is mimicry? Because mutation, guys, I thought of taking mutation today, but since we don't have a time, I'll take mutation maybe in the mutation in the molecular basis of inheritance. That time I take mutation. I just close it by mimicry. So because time is not favoring for us because we have another 5 minutes time. So I'm not doing mutation now. Mutation, the point mutation, gene mutation, frame shift mutation, insertion, deletion, translocation, transversion, all these things that we discussed when we start with the chapter, molecular basis of inheritance. Okay, that will do. So separately in that chapter, now we're not discussing about mutation. Okay, so I just close with the mimicry. Okay, this is the last part. The mimicry is also again a kind of the natural selection. So this was first discovered by Henry Bates, I think from the year 1862. 1862. The mimicry are of three types actually. So protective mimicry, aggressive mimicry, protective, aggressive mimicry, and we have a conscious mimicry. We have a conscious mimicry. So these are the mimicry of the three types actually. So how it mimics to protect it from the enemies, they protect from the other unknown species, all the organisms to safeguard itself from the predators. This use the mechanisms called process called as mimicry, right? Protective mimicry. The protective mimicry usually will be having two types again. Concealing mimicry. Just the names I'm giving you. I'm not going in detailed picture of this. And we have a warning mimicry. Warning mimicry. Protective mimicry are of two types. Concealing mimicry, uh, and we have a warning mimicry. So for example, you take concealing mimicry in case of praying mantis. Okay, praying mantis and warning mimicry. So warning mimicry you take in a poisonous, like sometimes like few species which will be like mimic like that of the poisonous coral snakes. That is like warning mimicry. Even though it is not poisonous, it behaves as if it is a poisonous snakes. So here warning mimicry, aggressive mimicry. Aggressive mimicry or which resembles like the color of the flowers. Sometimes the color of the flowers, it changes its color, camouflage with the color of the flowers. So this is type of aggressive mimicry takes place. Conscious mimicry, it behaves as if it is dead. Behave even though it is alive. Behave as if it's dead. It is dead. Okay. So this is just a piece of information that I given for you. Okay, so I done with this part, guys. So evolution part has got over. So if you have any questions, I have given 2-3 minutes time for you. If you have any questions, because few wants to ask some questions. You can ask your questions, so that by another 3 4 minutes I'll wind up the session. By this, we are completing this chapter, the evolution chapter. The only part is pending, that is mutation theory, that is Hugo de Vries. This alone we'll discuss maybe in the molecular basis of inheritance. That area will cover this particular part, that is mutation. Okay, so this chapter is over. Next class will start with human health and diseases. Human health and disease will start, and we'll try to complete the chapter. In human health and disease, we'll discuss first about immunology. Immunology will discuss, we discuss about the types of immunity, that is natural, artificial, all these things. Okay, I'll just show the human evolution slide. You want me to show the human evolution slide? Hey guys, this is also very important, guys. I forgot to show this one. This is already there in a textbook. The Silophyton, this area, because sometimes you get a questions which is a common ancestor for ferns, which is a common ancestor for conifers, which is the common ancestors for cycads. So this already there in your NCERT textbook. Please go through this diagram. Just one segment they'll ask. Okay, so this area you should be very careful. Again, this type of question, Permian period, Triassic period, Jurassic period. These two diagrams, don't think like you get not get questions. Sometimes the question paper is tough, then you cannot get any one segment of this particular diagram. You may get the question. So like this, we're done. Just overview of all these things. So which image you asked about? Okay, these are the different the images. Our ancestors, okay, our ancestors like Australopithecus, Homo erectus, Homo sapiens, Neanderthals, all these people. Listen, they are new one for us. They are all our ancestors only. Okay. So even though this chapter is very small, guys, but still two questions you can expect from this chapter, maybe from any area if you clear with this, but human evolution alone, try to grab as per as the information. SARS are in the same family. Why do they should use the treatment of SARS to cure COVID? But the structure, right? Even though they belong to the same, you take the answer retrovirus also. Retrovirus also, we have different types of retrovirus. But why we couldn't cure HIV virus, right? Where we can cure, perhaps we can cure other types of retrovirus, but why we cannot cure HIV virus? Where it gets hosted inside. This we call it a cell tropism. But when we talk about COVID-19, the cell tropism, it keeps on changing its shape. That is the one feature. Again, the characters, the proteins which present on the surface of the coronavirus, again, it's not like same like that of the other viruses. The genetic material may be like that of RNA. This might be the same like that of SARS virus, but the glycoproteins, the surface proteins, right, the cell tropism, these all the again seems to be different. So let's say like an example, you take retrovirus also. Other retrovirus we can cure, but why we cannot cure HIV virus? The same thing that why we cannot do it for the corona also, even though this belongs to the SARS. So of course, it comes under the SARS only. Beta coronavirus, we can talk about. But there are other mechanisms, the other structural protein seems to be entirely different. Sometimes it may be pleomorphic also. So again, there's nothing is impossible. Nothing is impossible. Still, our scientists are scratching our heads. We are still in the trial period, right? So hopefully, we find a new drug or vaccine for this particular corona. We'll come with a solution, and we try to destroy this virus that is conquering our world and the nation. We definitely will do it. Okay, so that will do it. Any other question? Grandfathers? Yes, Abhijit. So it's just check with the similarity of our grandfathers might be having loss of smell and taste as symptom of corona. Maybe not, because fever, a normal fever itself you take. If a normal fever, normal cold, if you take also you may not be having a taste. I mean the smell also. That is the usual common symptoms for common cold itself. That is the character feature of any fever, anything that you can get it, right? So it's not only for the coronavirus. So don't think like if you get a fever, you don't have a taste, you don't have a smell. The common cold, the nose blockage, the nasal blockage also, you get the same thing. What's your question, Raja Vignesh? What's your question? Because it changed time to time. Yes, that's what I told you. Pleomorphic. It keeps on changing. So like an HIV virus also, it keeps on changing its structure. It's a WBC. It's hosted in the WBC. The WBC, it's pleomorphic. It changed. It's today we design one drug. After we administer again, it changes its structure and shape. So that is actually the problem in this type of virus, which keeps on changing its shape. Affect the reproductive organs. So we're not sure about how it affects the reproductive organs. So far, so not at confirmed, no confirmed cases, right? If it, I may not say no also. If there are any few researchers which come up with possibilities where the people has got greatly affected with reproductive organs, there might be some chances. But as of now, not of much of cases has been reported where the reproductive organs been affected. Medicine to cure corona, then many of them are getting treatment. And also what this is a malarial drug. I told you at the time of the coronavirus, we are giving the malarial drug, we're giving HCQ, that hydrochloroquine, and that we're giving, and we're giving painkillers, the normal, the paracetamol kind of tablets only that we're giving. We're not giving actually any treatment, specific treatment for coronavirus. We are giving HCQ, malarial drug only we are giving as of now, and some viral drug also that we're giving. That is not selective with coronavirus. That's the reason why, we're asking the persons, infected persons, to be isolated at home itself, self-medication, isolating themselves at home, taking good rest, having food and sleep, taking proper rest, isolating themselves itself is a good cure. They can get cure of themselves in the early stages. Only in advanced stages only they should go to the hospitals. They should be under the supervision of the doctors or the providers. Then they need special attention for that. The initial stage, that is the reason why the government also telling me not come to the hospital, not get admitted. Be at home safe, be isolated, sleep well, eat well, take care of yourself. That is the best medication that you can give. It's like a common cold, and that's initial stage. If you identify the symptoms, you be at home, safeguard yourself, you'll get cured, right? So that is all in your mind only. Like, so you not get panic. It's like common cold, like this. It's like a common cold only. So only it's become like spreading outside. Only it's giving so many complications among ourselves. It's used to cure so that we can continue. Yeah, of course we're doing a malaria drug. Somehow it's giving, it's responding. So researchers are going on with this the malaria drug HCQ, that is hydrochloroquine, right? With this drug only, the trials are going on. Many patients started recovering, and certain places where the patients are not, the body is not responding to this particular antibiotic. So that is the reason why we are not prescribing this malaria drug to this type of patients. So that is the reason. So I'm not telling many, some patients are getting cured, but not the cure. The cure rate is not 100%. Okay, so what's the question, Kavita? You have some question. In morphology of flowering plants, you told that gamo means united, poly means free, but it's given that gamocephalus is check it out. Poly is no. Gamo means poly means, I think it's free. Can you please confirm it and tell me? So poly means free and gamo means united. That is what actually my reference says. Okay, can you just confirm and tell me, Kavita, so that it'll be easy for you. Okay, okay, guys. So your time is 11 o'clock, because you have the next session. The other faculty is also waiting for you. So every day we'll have this last 5 minutes session for you, so that you can ask questions, so that I am happy to clarify all your doubts. Okay, speciation originated from same population. It's not compulsory. It's not compulsory. It should be occur only with the same population. The different population also the speciation occur, but possibilities are more only in the same population. But large population or small population, that is matters a lot. If it is large population, no much of effects. Like that is the drift, the speciation occurs in small population, then abrupt changes will be more. That is the only thing that we need to consideration. But it occurs both in same population or different population. All right, guys. So thank you so much. So time is up. So 11:03. Every day we'll have this type of discussion. If you have any doubts in any of the chapter, you please make note of it. So I'll just give the last 5 minutes time for you every day, so that we'll have a discussion, any topics, and I mean any topics in biology, that is botany or zoology. So we'll discuss it. Okay. And Kavita, you have said like gamocephalus and poly, that one, right? So can you please just confirm it? Okay. Okay. And even I just confirm it on my side, so that I let you know in the next class. Thank you so much. Thank you. Have a great day. Take care. Thank you.